Let's start with a little review. So, how would you classify a bacterial species that grows only in the highlighted area of this culture tube? So remember, this is an oxygen gradient. You can see high, low, and no oxygen. We're looking at this area in blue here. Would you describe that as something that could only grow in there? Hint, hint. Would it be aerotolerant, a strict aerobe, a strict anaerobe, or a facultative anaerobe? Pause the video, think about that for a moment. All right, that would be a strict anaerobe because it can only grow there. Aerotolerant and facultative could grow there, but they could also grow in other parts of the tube. Strict anaerobes would only be able to grow down here. All right, so we tend to think of microbes as kind of singular entities, but they do work together, and in some cases they do form slightly different cell structures when they divide. So that process is called differentiation. Your body cells go through differentiation. You start as a single cell, a little zygote, and you become all these different types of cells. You got eyeball cells and skin cells and liver cells and brain cells. That's called differentiation. Microbes rarely will do that. So we'll talk about that. We're going to talk a lot about an important structure that can form called a biofilm. This is really critical for pathogens, but also environmental microbes to allow them to stay in locations and protect themselves. And when they're doing this, they can actually talk to one another. So there are communities of microbes that do communicate through this process called quorum sensing. We'll talk about biofilms in relation to infections primarily. And then we'll talk about sporulation, so forming spores and how that can impact different infections. Okay, so we are probably all familiar with biofilms. You just might not have called them that. A biofilm is a mass of bacteria that stick to and multiply on a solid surface. It could be a single species, or it could be a collaboration of multiple species, and they can stick to organic or inorganic surfaces, so they can form in your body. Biofilms are really disastrous when they form on things like urinary catheters. That causes a lot of hospital-acquired infections, and they can form in the bottom of ponds. The scum on the rocks and things like that can be biofilms on inorganic surfaces. And you're probably familiar with the common biofilm called plaque. Hopefully, your situation doesn't look like this, but you can see this bright yellowish material here. That is all microbial growth, and we call that plaque on our teeth. So what makes a biofilm kind of unique is that the microbes come together and they excrete what we call an extracellular polymeric substance, or EPS. This EPS is like a sticky goop that surrounds the bacteria. So here's like a electron micrograph of it. You can see all this brown stuff here. That's the EPS, and all these microbes are stuck into it. So this EPS helps bind them to a surface. In this case, they're binding to the teeth surface. So brushing is a way to try and break that off, right? And the thing that's unique about these biofilms is that not only do they help them stick, but they form like a protective coating. So this is resistant to chemicals, antibiotics, your immune system. It helps the microbes hide from that. So biofilms in infections can be incredibly deadly because they're shielded from antibiotics that we would use to treat them. The formation of a biofilm starts with what we call planktonic cells. Those are kind of freely swimming around until they find a surface that seems like it's good. They kind of start to attach to it, and they form these little tiny colonies, and they kind of twitch around and clump together, and then they start to secrete the extracellular polymeric substance, the EPS, and then they can form these giant towers here. And they start communicating through chemical messengers, and they're saying, hey, I'm here, you're there. And at some point, the density of the biofilm gets too much for the environment, and they talk to each other and they go, all right, we're going to release some of you, and you're going to go off and find your other location. These biofilm towers, like I said, super resistant to destruction. They're hard to get off. They're super sticky, and they're resistant to antibiotics. So let's go ahead and watch an animation about biofilm formation.
Bacteria were once thought of as unicellular organisms incapable of differentiation or communication. But we now know that many, if not most, bacteria form specialized surface-attached communities called biofilms. Indeed, within aquatic environments, bacteria are found mainly associated with surfaces. This biofilm is a plaque that forms on the surface of a tooth. Plaque is a mixed species biofilm, but single species biofilms also form in nature. For example, single species biofilms of Pseudomonas aeruginosa can form on the lungs of cystic fibrosis patients. The first stage of biofilm formation occurs when a specific environmental signal triggers unattached planktonic cells to attach to nearby inanimate surfaces by means of flagella, pili, lipopolysaccharides, or other cell surface appendages. The attached cells begin to coat that surface with an organic monolayer of polysaccharides or glycoproteins, to which more planktonic cells can attach. As the cells enter into biofilm formation, they no longer maintain their flagella, and instead may move along surfaces using a twitching motility that involves the extension and retraction of a specific type of pilus. Ultimately, they stop moving and firmly attach to the surface. As more and more cells bind to the surface and divide, they form microcolonies and can begin to communicate with each other by sending and receiving chemical signals in a process called quorum sensing. These chemical signals are continually made and secreted by individual cells. Once a population reaches a certain number, analogous to an organizational quorum, the chemical signal reaches a specific concentration that the cells can sense. This concentration triggers genetically regulated changes that cause cells to bind tenaciously to the substrate and to each other. Quorum sensing may influence the amount of extracellular matrix produced by the cells. This thick, slimy material consists of polysaccharide polymers, called exopolysaccharides, and entrapped organic and inorganic materials. These exopolysaccharides increase the antibiotic resistance of residents within the biofilm by limiting antibiotic access to the biofilm center. The chemical signal molecules also stimulate production of some antibiotic resistance mechanisms. As the biofilm matures, the amalgam of adherent bacteria and matrix forms columns and streamers, creating channels through which nutrients flow. Although most of the bacteria adhere to the biofilm, some planktonic cells leave for new environments. Here is an image of the lumpy, uneven biofilms created by a mucoid environmental strain of P. aeruginosa. Biofilms allow bacteria to stay where food is plentiful. Why should a microbe travel off to hunt for food when it's already available? Once nutrients become scarce, however, individuals will detach from the community to forage for new sources of nutrients. P. aeruginosa produces an enzyme that can strip away the exopolysaccharides and will resynthesize flagella. Cells leaving the biofilm revert to their flagellated planktonic forms and can swim to new locations where nutrients are more plentiful. A hallmark of infectious diseases in which biofilms play a prominent role is their chronic nature. A biofilm infection may linger for months, years, or even a lifetime.
All right, so biofilms, medically, pretty nasty things to deal with. We saw that example, cystic fibrosis. The lungs have buildup of material in there. Also, urinary catheters tend to see biofilm formation on them. So they can become difficult to treat when that happens because they're resistant. Another structure that microbes can make is called the endospore, and we've talked a little bit about this previously. but this is more like a dormancy structure. So certain gram-positive bacteria can produce highly resistant and strong endospores, they're called. So we've seen this previously with Clostridium. We saw Clostridium botulinum in the chili can example. Tetanus bacteria, Clostridium tetani, can form it as well. Also bacillus, such as Bacillus anthracis, which can cause anthrax poisoning. They make spores. So when the bacteria go into a spore, they're dormant and they require no nutrition or energy at all. And this can stay dormant for decades, hundreds of years, and in a couple of cases, we have found spores basically preserved in amber on insects, and they were actually able to extract the endospores from them and germinate them from 25 to 40 million year old endospores from a bee gut in amber. So it's not Jurassic Park. This is the Paleogene era. So it's Paleogene Park, and it's not a T-Rex. It's spore forming bacteria. So it's not quite as exciting. No dinosaurs walking over your head, but still really, really cool. If you want to see the publication on that, it's here. Spore formation is a process of asymmetrical cell division. So normally we have what's called vegetative growth, where the cell reproduces its genome, divides and septates, and now we have two identical daughter cells. When a microbe senses that the environment is going south, right? It's too hot here. It's too dry. There's things attacking me. It can switch over to what's called sporulation. So we're going to follow this around. So it starts duplicating its genome, and it makes an asymmetrical division where it starts pumping some of the genome into it. This is called the forespore. The original cell is called the mother cell. And then it starts kind of enclosing and encapsulating the forespore. So it creates a protective layer around it. And further layers are added: the cortex, which is some extra peptidoglycan, and then the exosporangium, which is really resistant layer. And then the mother cell starts to break down. And the spore coat is added, because it thinks calcium is in there. And finally, the original mother cell gets broken down, and we release that mature spore, which is highly resistant to desiccation or drying out, highly resistant to heat. That's dangerous because we might treat things with like boiling. Like if you have a boil water order, you treat water by boiling it, but these resist boiling. They can resist chemicals, they can resist antibiotics. The only way to really destroy spores is heavy bombardment with high energy radiation, X-rays, gamma rays, or autoclaving will destroy spores if you do the autoclave run properly. So that's hard to do on a lot of substances though. So spores, they can be dangerous, and historically, anthrax, Bacillus anthracis, has been a livestock disease because the spores, they stay dormant in the soil, and livestock would go out and they paw at the dirt and eat and things like that and inhale the spores, and then they go into the lungs where they start to germinate, and then they can cause disease in there, which is anthrax disease. Now, you probably know anthrax as a bioterrorism agent. It's so effective for bioterrorism because it makes these resistant spores, so you could ship it in the mail, and unless our mail gets irradiated like it does now because we've had anthrax attacks, it can survive, just shipping around unless you irradiate the mail. Okay, let's watch the animation of endospore formation.
When starved for nutrients, certain gram-positive genera, including some important pathogens, can develop dormant heat and desiccation-resistant spores. Most of our knowledge of bacterial sporulation comes from the gram-positive soil bacterium Bacillus subtilis. When growing in rich media, this microbe undergoes normal vegetative growth. B. subtilis can replicate every 30 to 60 minutes. In contrast, starvation initiates sporulation. an elaborate 8-hour genetic program that ultimately yields a spore. In the first stage of sporulation, the septum forms near one pole of the cell rather than in the middle. The DNA replicates and extends into an axial filament that spans the cell's length. During the next stage, the septum separates the forespore, which will ultimately become the spore, from the larger mother cell. DNA is pumped through the septum until each compartment gets a chromosome. The mother cell now engulfs the forespore, surrounding it with a second membrane. The mother cell chromosome is eventually destroyed. A cortex, made up of a thick peptidoglycan layer, is placed between the two membranes surrounding the forespore. This cortex will be important in providing heat resistance and maintaining dormancy to the spore. Layers of coat proteins are deposited on the outer membrane. In the next stage, dipicolinic acid and calcium are incorporated into the spore. Dipicolinic acid is important in maintaining dormancy and in promoting spore dehydration, a characteristic that makes a spore heat resistant. The mother cell, now called a sporangium, dies and releases the mature spore. Spores are resistant to many environmental stresses that would kill vegetative cells. The nature of this resistance is due in part to the spores having only 10 to 30% of a vegetative cell's water content. But spores also contain small, acid-soluble proteins that bind to and protect DNA from damage by ultraviolet light and various toxic chemicals. Fully mature spores can exist in soil for at least 50 to 100 years and have been known to last thousands and even millions of years. Once proper nutrient conditions arise, another genetic program, called germination, is triggered to wake the dormant cell, dissolve the spore coat, and release a viable vegetative cell.
All right, so endospores, of concern for bioterrorism reasons, medically of concern because they're very difficult to deal with. The only way to get rid of them is autoclaving or irradiation, which we can't really do to everything. So cleaning spores is difficult. All right, that finishes up Chapter 6.6 and Chapter 6 as a whole. We talked about two structures that can form here: biofilms, multicellular surface-attached microbial communities, that extracellular polymeric substance there. In there, bacteria can communicate or quorum sensing with one another, to talk with one another to figure out how dense to make the biofilm. And this sticky substance is of concern to us because it sticks to surfaces that we try to clean, but also it's resistant to chemicals and antibiotics. And then we have endospore development in some of our gram-positives, Bacillus and Clostridium, that are of concern because they're dormancy structures, so they can stay and wait and hide, basically, and they're resistant to drying out and heat and chemicals and things like that. So medically, very interesting. Also, for livestock, they're of importance too. Okay, that ends Chapter 6.